Hello everyone, this is the first of a series of videos dedicated to organic chemistry in the context of biological systems. We are going to be looking at some general concepts related to organic chemistry and specifically bioorganic chemistry, organic chemistry in biomolecules and biological systems. We will describe what isomerism is, types of isomerisms, and the role of stereochemistry in the context of biological systems. We will also be learning about general reaction mechanisms, what's a nucleophile, an electrophile, a living group, and we will study specific reaction mechanisms, nucleophilic substitutions, oxidative additions and reductive eliminations, electrophilic additions, and electrophilic aromatic substitution. We will also learn how to predict the products of two of these types of reactions, specifically nucleophilic substitutions and elimination reactions. And finally, we will be looking at the role of these general reaction mechanisms to describe reactivity in biological systems. As usual, this topic is separated in several videos to make it easier to follow. So, what is bioorganic chemistry? Well, we first need to start defining what organic chemistry is. Organic chemistry is the branch of chemistry that studies carbon-based compounds. And it looks at the structure and the design of those compounds and the interaction between the different structures and how the structure of a compound impacts its function. And it also looks at synthesis, how to produce those organic compounds. And sometimes those synthetic routes are produce analog products to natural products. And there's also an aspect of organic chemistry that looks at kinetics, at the details of the reaction mechanism, and it covers the quantitation of those reaction mechanisms, how much product is produced and how fast the product is produced, and how to improve the speed of that particular reaction. Bioorganic chemistry is the branch of chemistry that uses the principles and tools of organic chemistry specifically to the biological context in understanding biochemical and biophysical processes. In the next few slides, I'm going to have a brief overview of some key concepts in organic chemistry. When we look at an organic compound, we looked at the backbone of the organic compound and the functional groups of that compound. The backbone is the skeleton of carbon hydrogens that make the basis of that compound, which is usually unreactive. And then in that carbon hydrogen skeleton, there are a series, one or more functional groups. And those are groups, a specific combination of atoms that repeat over and over again throughout organic chemistry that display particular characteristics, particular chemical and physical properties. And typically those functional groups are polar. And you have here in this example some examples of functional groups from double bonds, a hydroxyl group, an amino group, a carboxylic acid, carboxylate group, or an amide groups. You can classify organic compounds according to their backbone, according to their carbon chain. The first step in the classification would be to separate them into acyclic and cyclic compounds. Acyclic compounds can be saturated, so no double or triple bonds, or unsaturated with at least one or more double or triple bonds. If you have a cyclic compound, you typically have a carbocyclic compound, and that is a cyclic compound exclusively made of carbon atoms. The cycle is exclusively made of carbon atoms, and you have here the example of cyclobutane, and here the example of benzene, in which the cycle is in both cases exclusively made of carbon atoms. And then within those carbocyclic compounds, they can be alicyclic, which means single bonds, or aromatic, which means they have alternated double bonds. And your cyclic compound can also be heterocyclic, and that means one or more of those positions in the cycle are occupied by an atom that is not carbon. Here, for example, we've got an example of two different cycles, both with one of the positions occupied by a nitrogen. And within that heterocyclic compound, you can have non-aromatic or aromatic cycles. And here, for example, this is the example of pyridine, which is an aromatic compound, an aromatic cycle, in which one of the positions of the cycle is occupied by a nitrogen. You can also classify an organic compound with respect to their functional group, their key functional group in the molecule. And you can have halogens as functional groups. You can have a hydroxyl group, and that gives you alcohols or phenols if they are in an aromatic ring. You can have ethers, ethiols, and sulfides when you have sulfur in the structure. And here just some examples of how those organic compounds might look like with those particular functional groups. More examples of how you can classify an organic compound with respect to its main functional group. Some of them hopefully sound familiar to you. You've got here the amino group and the amines, or the carbonyl group that gives you aldehydes and ketones, or the carboxylic acid group that gives you carboxylic acids. And you also have other groups that maybe are less familiar to you. For example, the sulfur group and the sulfonic acids, which we will see can be used in future years. You will see how they can be used as protective groups in synthetic chemistry. Or, for example, the nitro group and the nitro compounds. And we will study in future years the use of nitro compounds and nitro groups as good intermediate in reactions. Again, something that hopefully will sound familiar. When you write an organic structure, you can write the condensed formula, which is just the list of the different atoms that make part of your compound in one line. You can have what we call the semi-dash structure, in which you are having a 2D representation of your compound. You still show all the atoms, but you're also trying to show in the structure the shape, the three-dimensional structure of that particular compound. Or finally, a very, very common way of representing it, and it's the way you will find it both in papers and in chemistry books, the bond line formulas, in which you only show atoms that are not carbon and hydrogens. And as you can see, the skeletal form, the carbon backbone, is completely implicit. You don't have any carbons or any hydrogens, just the lines that represent those carbons and the hydrogens. And the only atom that has survived is the chlorine, because it's not a carbon or a hydrogen. A carbon involved only in single bonds, an sp3 carbon, has a tetrahedral structure, and the substituents point towards the corners of a tetrahedron. Sometimes we can try to reflect that three-dimensional structure when we're writing our compound in a 2D structure, and for that we use the wedge bonds and the hash wedge bonds. And what they are in trying to represent, the wedge bonds, are that particular substituent, that particular atom, that particular part of the molecule is pointing towards you. And when you use a hash wedge bond, what is representing is that particular atom or section of the molecule attached to that hash wedge bond is pointing away from you.